everybody. Today we're going to talk about nervous systems. Now, when you think nervous system, we typically don't think about lightning bolts coming from and to our heads. But if we really take a look at the science that's going on, what we find is that there is little bits of electricity that are involved with how one neuron communicates with another. And that's what we're going to delve into today, how neurons communicate with each other. Now, spending just a few moments on some of the simpler animals, we can first look at the hydra. The hydra is commonly looked at as a simple nervous system because it's nothing more than a nerve net. A nerve net kind of is what it sounds like. It's just a random assortment of what look like fibers passing through the body and the tentacles of this little aquatic animal. Now, there's no accumulation of cells anywhere that we could call a primitive brain, but this nerve net, these neurons, do respond to stimuli, and they also are able to coordinate and capture prey, and that's because they're in contact with contractile epitheliomuscular cells. So this little tiny critter has to have a means to feed itself, and it does so through its nerve net. Second, we can take a look at the planarian. The planarian right in the middle here, you can see that it's a little bit more than a nerve net. You have a couple of tracts, one on the left and one on the right, but then we also do see an accumulation of nerve cells here that we're going to call ganglia. We're not going to go as far as to call it a complete brain, but we will call it a ganglia, an accumulation of nerve cells. It also has an eye spot and an auricle. So these are sense organs. So we've just taken the hydra's nerve net and we've just fast forwarded it into complexity a bit by having accumulation of nerve cells that do a little bit more than just responding blindly to stimuli. So what we can say then is that one specific trend in the development of nervous system is going to be bilateral symmetry. Now, that's evident on the planarian because if we split this thing down the middle, it has a right side and it has a left side, and they tend to be mirror images of each other. Another thing we can throw onto that idea is the idea of cephalization, or the accumulation of more development of the nervous system towards the head of the organism. If we look one step further at the earthworm, you can see there's actually something that will qualify as a brain. So a little bit more than the planarian. Earthworm is quite a bit larger than the planarian, and the nervous system is a little bit more developed. In the case of, instead of ganglia, it now has an accumulation of enough tissue to call it a primitive brain. So as we go further in complexity, we eventually reach the vertebrate nervous system. Vertebrate nervous systems are composed of two things, brains and spinal cords. So the whole cephalization idea that you'll have an accumulation of nerve cells at one end of the organism is firmly present in a human. And we also have a cord coming from that cephalization that's going to allow us to then have effectors. We're going to have organs and body parts that can move that are connected to that brain. So this comes from embryonic tissue called a dorsal tubular nerve cord. We have it right here. So early on in development, what we see are a couple of folds in the head region of organisms, and those folds over time will develop into our brain and spinal cord. Another thing that we see with vertebrate organization of the nervous system is we see paired sensory receptors. So our eyes, our ears, and of course our nasal passages. We have two. So we tend to see that in higher organisms as well. Here, for example, is these are the cranial nerves of the frog that you see right here. And notice that everything has a left side and a right side, so it's doubled up. So an olfactory nerve, this one here, is going to control the frog's ability to smell. Here we have a couple of optic lobes, one on each side. Frog obviously has two eyes as well. And you can see everything kind of split right down the middle, doubled up. And that is another thing that develops in higher organisms versus simpler ones. Organization of the vertebrate brain has three major parts. The first part of a vertebrate brain is the forebrain, which you can see here. The forebrain is usually composed of quite a few things, but the two most typical, most prevalent in a vertebrate are a cerebrum and an olfactory bulb, which is going to enhance the ability to smell. Now, the second part in a vertebrate brain that seems to always be there amongst different species is the midbrain portion, which here we'll just kind of circle this centermost area, and it's composed of a thalamus, which you can think of kind of as like a relay station for messages both going up and down, a hypothalamus, which sits below, and that's where this hypo prefix comes from, and this is kind of like your brain's pharmacy. It's right here. And then optic lobe as well. Most vertebrates have the ability to see. And we also have a hindbrain. And the hindbrain is the portion of the brain that deals with the regulatory stuff that keeps you alive and conscious of your surroundings and all of those survival type mechanisms that are inherent to vertebrates. Now, if we go ahead and we throw a human brain up there, are those parts there in our brain? Of course, we're vertebrates too. What we can do is we can kind of just give you a little outline of where these things are at. So if we follow along right about here, we go from forebrain to midbrain to hindbrain down here. Now, what's all this extra stuff? Well, that's with the bells and whistles we were talking about. So we have a lot going on in our brains, and actually very large for our human size compared to a lot of other species in the size of their brains. Alright, so we can break the human nervous system down, at least in the beginning here, into two parts. One is your central nervous system, also called a CNS. So it's your structural and functional center of your entire nervous system. It's composed of your brain, your spinal cord. It receives sensory information. It integrates it, evaluates it, and then initiates an outgoing response. So if I have a sensory type of stimulus that hits my hand, that's going to travel up my hand, eventually reaches my spinal cord. It's going to be processed in my brain up here, evaluated, and it's going to initiate some kind of response. Maybe my hand is uncomfortable, so it's going to propagate another message going the other way to have my hand move. Now, the other big partition from the CNS is what we call our peripheral nervous system, or our PNS. So this consists of nerves that lie in the outer regions of our nervous system. Now, when we first sensed our hand was uncomfortable, it was through stimuli that eventually and essentially went upwards. Now, that is what's considered a peripheral nerve because it starts outside your nervous system and it's going to bring a message into the central nervous system. Whereas when we had the outgoing message, which was coming from our brain and it was going to move down in terms of, we call that a motor type of movement, that one is going to be from your central nervous system because it originated in your brain and it ended in your effector organ. In this case, it happened to be your hand. So we have the CNS then, and then we have the PNS. CNS has neurons that originate in the brain and spinal cord, and your peripheral nervous system has neurons that originate from the outside and go towards that central nervous system. Now, within the CNS, we also have some very famous nerves. We have what are called cranial nerves, and we have what's called spinal nerves. So cranial nerves, of course, they're going to originate from the brain, and they're going to move outward from the center into our various senses. And spinal nerves are going to emerge from the spinal cord, and they're going to head outward in the body to innervate a particular organ or body part. If we take a closer look at those cranial nerves, what we have here, if you look at, you'll notice how all of these little tiny, what look like yellow tubes come to an abrupt end. Well, that's just cut for this picture's sake, but these are our cranial nerves here. These are going to control our senses, and there happen to be 12 of them. And if you take anatomy and physiology, most likely you'll have to learn all 12 of those, and you're going to learn what they do, you're going to learn how they get their sensory information, and then how they send out their effector messages. Over here to the right, we see a spinal nerve, and we have a lot of these. We have quite a few pairs that are going to go in between each vertebrae through a dorsal root, and each dorsal root also has a dorsal root ganglion. So we said earlier, a ganglion was a clump of nerve cells together. Now, messages that are coming in are going to come in through that dorsal root ganglion, and they're going to find their way into the spinal cord. Now, whether it's a reflex action or whether it's an action that can also require thought will determine how fast the effector message goes out. And the effector message goes out through the front hand side and through what we call the ventral root. And also, at the same time, we're probably going to have a message reach our brain sometime later if it's a reflexive type of stimulus. Down here is what it really looks like if you take a look at a cross-section of the spinal cord. It has a central canal with cerebrospinal fluid. There's gray matter, which we'll understand what that is in a little bit, and then there's white matter on the outside. So it kind of looks like an H or the outline of a bat on the inside. All right, now we have also a system called a somatic system, and that's part of the PNS. So what that's going to do is it's going to handle sensory information from things like your skin, your joints, and your skeletal muscles. Those are going to send signals to your central nervous system through your peripheral nervous system. So they are a part of the peripheral nervous system. And those messages that we're talking about are coming from what we call the afferent division. It consists of all incoming impulses, also considered sensory. And secondly, the efferent division consists of all outgoing impulses. So those are what we call motor. Now, how do you remember those things? Well, most people will go ahead and they will use the acronym SAME, S-A-M-E. So here we have sensory, and that happens to be the afferent pathway. And here we have motor, and that is the efferent pathway. So if you're having a hard time remembering it, go with the SA combo and the ME combo in the word same. Okay, afferent division and efferent division is how we can also functionally classify the nervous system. It consists of messages that are coming in and messages that are going out. Here's an example of what we're talking about. So we have receptors in our skin. So right here, let's say we sense hot, cold, or pressure, and that's going to send a message through the afferent neuron. Here happens to be the cell body of that afferent neuron, and it's going to go all the way to our spinal cord. Well, that's a long neuron. Absolutely is. So you have some neurons that travel very far. Some can be up to 3 feet in length. Then it's going to reach what's called an interneuron. More on that in a little bit later, but it's basically a neuron that's found entirely within the spinal cord. And then it's going to butt up next to what we call the efferent or the motor neuron, which is then going to initiate a response, again another very long nerve cell, in this case to an effector, which is a muscle. If we talk about sensory pathways and motor pathways, we have to mention what a reflex arc is. It is a hardwired, unconscious rapid response to external stimuli. This involves sensory neurons, interneurons found entirely within the spinal cord, and motor neurons. So again, this is unconscious. You will respond to certain stimuli before you even think about that stimuli. And that's one of the things that keeps us safe. So the thought comes later, but the reaction comes first. So let's take a look at a scenario that utilizes a reflex arc. This comes from the website Sumanis Inc. It's one of my favorite in terms of the animations they have. So I will narrate for you as we go through the information. The speed of information processing partially depends on the number of neurons involved in the task. A complex voluntary behavior, such as identifying and reaching for an apple, involves multiple brain systems, sensory, cognitive, and motor, and many neural connections as well. The elapsed time between recognizing the object and initiating a behavior is thus relatively strong. While complex, voluntary behaviors are certainly adaptive. There are situations such as the rapid withdrawal from dangerous stimuli that require immediate action. A rapid response is one of the advantages of the reflex arc, which only involves a few neurons. In a simple reflex, the entire signaling sequence, detection, processing and response is handled by the spinal cord without the need of input from higher brain regions. Indeed, conscious perception occurs after the reflexive withdrawal has already begun. Let's examine the reflex arc in more detail. First, specialized sensory fibers in the skin detect the painful stimulus and relay this information to nerve cells in the spinal cord. The information is carried all the way from the skin to the spinal cord by a single sensory neuron without any intervening synaptic connections. There's our stimulus. You see it traveling up to the spinal cord. Once in the spinal cord, we'll see what happens next. The sensory fibers enter the spinal cord and synapse amongst interneurons within the spinal cord gray matter. These interneurons, in turn, synapse upon the motor neurons that connect the skeletal muscle. In this case, the flexor muscles in front of the arm are constructed to contract, while the extensor muscles in the back of the arm relax. So what we see here is we see that the arm flexes in response to the painful stimuli because it received a message from, in this case, the stimulus through the arm up into the spinal cord, and there it was acted upon by two interneurons. One of them was excitatory, the one on the left. One of them was inhibitory, the one on the right. By having an excitatory interneuron, that is going to stimulate this flexor muscle to contract. So her arm is going to pull away. And for that to happen, the tricep muscle cannot be flexed as well. If you want to try it right now, flex your bicep and try to flex your tricep at the same time. It's not easy to do. But to flex your bicep means that you are inhibiting your tricep. Your tricep is relaxing as your bicep contracts. And the same goes the other way. If you push your arm outward, you're going to feel your tricep contract, and then if you feel your bicep, it's going to be loose and it's going to be relaxing. So through exciting one neuron and inhibiting another neuron in the back of the arm itself pulls away from the threat, and milliseconds later your brain perceives what just happened. Pretty amazing system. Here's a still of a reflex arc, once again showing the path of a spinal reflex. So here we have very painful stimuli. We have a pin, which has now found its way into your skin. There are sensory receptors here, and there's a number of them, and this one happens to trigger off a pain message, and this one is going to then send its signal up through, in this case it will hang out for a second in the cell body before being continued to be passed on down to the interneuron. In this case, our interneuron is this big purple one in the center. And that interneuron will synapse with a motor neuron here, which is going to send the message to the bicep to flex because that painful stimuli must have happened in the forearm to cause that to occur. All right, so we've talked about various divisions of the human nervous system. We talked about how the central nervous system had the brain and spinal cord as part of it. Now, in the peripheral nervous system, we said those were originating from outside of the central nervous system, everywhere else in the body. And we have certain fibers, somatic sensory fibers, that come from skin and the special senses that we have. But we also need messages coming from our visceral sensory fibers. We have internal organs that need to send messages to the brain as well. And typically, these do so without any conscious control. They just kind of do everything on their own. So those are somatic, both of them being sensory, send information upward. So these are also afferent as well. Now, the messages that come with these down arrows here, first, somatic motor fibers, so these are part of our efferent system. These are sending messages outward, in this case to skeletal muscles to relax or contract. And we also have autonomic motor fibers, which are going to tell our internal organs what to do. Whether they should speed up, be more metabolically active, whatever the message may be, they're going to respond to it. And they're going to respond to a system that's broken into two categories. One is called the sympathetic division, and one is called the parasympathetic. So whether you've taken psychology or maybe taken an intro bio course, hopefully they covered the sympathetic, which is our fight or flight system. So those scenarios will provide a lot of stimuli that get someone very worked up in a very short amount of time. Often the case, they might be it might be in survival mode, whether it is appropriate for the scenario or not. Running from a bear in the woods, very appropriate to be in a fight or flight type of a body condition where every resource your body has is being spent to stay alive, and all of the unnecessary systems that we have are shut down because we want all the resources going to the things that will enhance our survival. Now, the parasympathetic division over here is also called the rest and digest. So this is where we want to be more of the time in our life, in a rest and digest mode, because it's very relaxing. It's when your body kind of slows itself down, and it works on all those things that it doesn't need to do during fight or flight, like digestion and lower blood pressure and all of the restorative processes that your body does. Now, there are three structures, key structures to any nervous system. First, they're made of excitable cells. They have to initiate and conduct impulses that make possible all nervous functions, and these cells are your neurons. Components of a neuron, there are three of them. We have the cell body, also called the soma, and this is the biggest part of the cell that looks pretty weird to us. Now, we're, in introductory biology, we always meet cells that look like big round blob, but this one definitely does not look like a big round blob. The only part that does is the cell body or soma, and the parts inside of it are very familiar to us: nucleus, cytoplasm, ribosomes, endoplasmic reticulum, Golgi apparatus. That's all there. What they provide: protein molecules called neurotransmitters. So each one of these is able to produce little tiny chemical substances that are going to have some impact on the way we think and the way that nerve impulses are fired, and are going to aid to our consciousness. How are these neurotransmitters made and passed on? They are packaged into what's called a vesicle. A vesicle is kind of like a vacuole that contains a whole bunch of chemical to be transmitted to another cell inside of a nervous system. The second part of a nerve cell is the dendrites. The dendrites come from the prefix D-E-N-D-R, and this, we're talking about branches. And if you look on the outside of that cell body right here, you can see a lot of branches. Almost looks like a tree that you might have in your front yard, where the main mass is in the center and there are various branches coming off the side, and as they poke further and further out away from the center line of the tree, they get smaller and smaller. And that's the essence of a dendrite. They are collecting branches, and what they're going to be collecting is nervous stimuli from other cells that are going to eventually, possibly, make this cell here fire as well. The distal end of dendrites are called stimulatory or excitable because they have little gates on the outside of them that respond to stimuli. Now, the stimuli can be excitable stimuli, or the stimuli can impair the ability for this nerve cell to fire. So instead of being kind of an excitable type of message, it might be a message that sends it away in a direction where it's not going to continue on the message. And we'll get more into that in a little bit. The third thing that all nerve cells have are axons. And axons are going to extend from what's called the axon hillock. And I'll go ahead and I'll outline that right here. Axon hillock is this region right here that comes from the cell body, and it is headed towards this long tentacle type of structure coming out of the cell body or soma, and this is your axon. Now, this axon here happens to be wrapped. We'll get into what these are in a little bit, but the idea is that the nerve signal is going to pass along the axon, eventually leading to the bottom here. So those little tiny things, you can see their name, they're called myelin sheaths, and there can be multiple myelin sheaths on an axon. The axon will conduct a nerve impulse away from the cell body. So what that means is that it's headed down this way. It collects from out here, processes in here, and then sends that excitable message down the axon towards these axon terminal knobs. These axon terminals are also called synaptic knobs as well, or sometimes I've heard of them as the terminus of a neuron because it's the end. They're also, if we could really zero in on one of these, we find what's called a presynaptic membrane. The very outside surface of each one of these synaptic knobs is going to be referred to as the presynaptic membrane. When we talk about the synapse, that's going to play a key part. So right here we have a generalized neuron, and we can see the direction of flow of messages when it receives them. So the messages are going to come in through the dendrites, and it's not just always one message. In fact, it virtually never is. Instead, it's a multitude of messages that are hitting different branches along these dendrites, and their collective input may propagate a nerve impulse to be continued, in this case from left to right. Second, we have the cell body, which contains all the organelles we learned about when we learned about the cell. It even has a nucleus right here. These messages will continue on this way and eventually all lead up here to this place called the axon hillock, which will determine if it's strong enough to keep it going down the axon here from left to right. So here's our axon. And this one here, it's not myelinated. We don't have it wrapped in those what look like little donuts around it. We'll talk about the difference between myelinated axons and unmyelinated axons in a little bit. So one of the things that I love to have my students do is this. They are to make a mad neuron. So if you type in to Google search or whatever your favorite search engine is, make a mad neuron, you can be Dr. Dendronstein. And that's this guy right here. And you can go ahead and put together and see if you remember all of the terminology for a nerve cell. Go ahead and give it a try. It's fun. Now, we talked about the wrappings of axons, and that's called myelin sheath. Now, myelin sheath producing cells in your peripheral nervous system, so cells that start outside the brain and spinal cord, they come from cells that are called Schwann cells. Their job is to wrap up the axons in myelin. Now, why would it do that? What's so important about myelin? Well, myelin is going to allow 10 times faster transmission of messages. And that's quite a bit faster. That's absolutely significant. So by wrapping an axon up, by some reason you'll see in a little bit, we're allowing a message to go much faster than it could an unmyelinated axon. Myelin-producing cells in the central nervous system, those that are found in the brain and the spinal cord, are called oligodendrocytes. They're little helper cells as well that are going to wrap those neurons in a myelin sheath. Now, the myelin sheath is kind of a fatty substance, and it's kind of, think of it like an insulator on top of like a copper wire. It has the same type of functionality. Now, you'll also notice that the myelin doesn't do a complete wrap. There's nodes along the way, and I'll circle them here. And these nodes allow for conduction of nerve impulses to happen. So if it was completely wrapped, we may not get the efficiency that we do with these multiple gaps that are there. And maybe it's just too much for a cell to wrap an entire axon because they tend to be very long. So we do see a lot of gappage going on along the way of an axon. Where you have these gaps, those are called nodes of Ranvier. And you'll see these when we discuss saltatory conduction. The nodes of Ranvier play a big part. Two neurons don't actually touch one another. Instead, there is a spot in between them called the synapse. And here you can see it as the gap that lies between these two cells. So the gap itself, the space between two neurons, is actually called the synapse. And as you can see here from the description, it's located at the junction of the synaptic knob of one neuron, and that's this right here. We talked about the terminus of an axon terminus, which is a synaptic knob. And here is where we're going to hopefully propagate a message to the next one. This could be the end of a branch of a dendrite or a nervous cell that's receiving the message. But again, this space right in between the two here, this is what we refer to as the synapse. Now, in this picture here, you can see that there's something going on between the two. And in this one here, it happens to be release of a chemical. Notice that on the surface of this neuron here, this is the dendrite that's catching the message. We see protein receptors. And this receiving membrane of a dendrite is called the postsynaptic neuron. Or you could even call it the postsynaptic membrane. And it has little protein receptors that are going to catch, and here comes another one, catch these neurotransmitters that are being released from the axon, the synaptic knob of the presynaptic membrane. So that would be the presynaptic membrane into the synapse. And then the postsynaptic membrane catches the chemicals and will transform it from chemical energy into electrical energy, and we'll learn how in just a little bit. Now, those neurons can't do it all by themselves. They need helpers. So the supporting cell cast of the nervous system is referred to as glia. Glia covers about 5 different types of cells that aid and assist neurons in getting done what needs to be done. The first one we'll mention are astrocytes. Astrocytes, they get their name from astro, which roughly translates into star. So they have a star-like shape. They're pretty large, and they're the most numerous types of glia. They have cell extensions that connect to both neurons and capillaries. So if they're connecting neurons and capillaries, that means that they are kind of like a go-between between the blood supply and those brain cells. They're very important. They transfer nutrients from the blood and the neurons. They pick up glucose from blood. Your brain has to run on some kind of sugar. They convert it to lactic acid and then pass it to those neurons. So they're a chemical intermediary between the two that are going to allow the glucose to be put into a usable form for your brain. They also form tight sheaths around those brain capillaries, which with tight junctions are going to constitute what's called the blood-brain barrier, the BBB. So they're going to prevent bad stuff from getting in your brain cells, as important as they are, and they're going to kind of work in terms of an overseeing type of functionality to make sure the right stuff is getting in the brain cells and preventing the wrong stuff from getting in there, providing a barrier. Second, we have little helper cells called microglia, and these are small cells that will respond to inflamed brain tissue and phagocytized threats. So here we kind of have a picture showing just about everything, but first thing I'll mention here, we'll go from left to right, we have oligodendrocytes. So that's this player right here, and this one is currently wrapping that axon, this axon, forms a second sheath around this one. So in some cases, it can form multiple wrappings around those axons of neurons. Here is the myelinated axon. You can see it's completely covered by myelin sheaths. Someone's cut off a bit here, so you can see the axon underneath it. Here is an astrocyte. This was the type of cell that allowed us to have a blood-brain barrier. And notice how it's gripping around the blood supply, and it's directly in between the neuron right here and the blood supply. So it's the go-between, and it's going to keep you safe from things that are in the blood, and it's also going to extract nutrients from the blood that your neurons need. We also see the microglia up here. These were the little helpers that are trying to keep your brain tissue safe, and they're going to phagocytize debris and things that are not supposed to be there. They'll respond to chemical messaging when there's damage and tend to congregate in that area. Last but not least, on here we have ependymal cells. These tend to line various ventricles and open spaces of cerebrospinal fluid in the brain. They actually produce the fluid, and here you can see some cilia on the outside of it. So they'll actually provide a little bit of a current in the cerebrospinal fluid as well, with the coordinated beating of that cilia. And here is a bunch of microglia that has been fluorescently enhanced in an image. Okay, keeping on the topic of microglia, notice here we have some resting microglia cells, not doing too much, hanging out for any signs of damage. But then right here it says a neighborhood in ALS. So ALS is also known as Lou Gehrig's disease, and what's going to happen from it is nerve cells in the nervous system, they're going to slowly degenerate over time, breaking the connection between the brain and the efferent nerve cells, the ones that are going to send messages to your muscles, your skeletal muscles. And unfortunately, there is no cure for Lou Gehrig's disease, but notice that the cells have activated the alarm system here, which are these inflammatory signals, which are going to activate these microglial cells, and they're going to start going and trying to find foreign debris and things that are damaged to phagocytize those things. Here you can see microglia all around, in this case the blood vessels as well, similar to how the astrocytes were in that same general area. And here we have, this is a nerve cell, and various branches coming to the cell body, and these are microglia that have been stained, so they're all around it at any given time, searching out trouble. We're now to the nerve impulse. Now, nerve impulses are electrical in nature. They're also ion-based. As you know from basic chemistry, ions are little charged particles, and they're going to play a part in this idea as well. So the electrical differences that we're going to find inside the brain from moment to moment, from cell to cell, are going to play a key part in the nerve impulse. The nerve impulse is also referred to as the action potential, which we'll get to in a minute. Alright, so let's start filling in some blanks. A wave of electrical fluctuation that typically travels along a neuron's membrane goes in the direction of dendrite to soma to axon. We covered that when we looked at the nerve cell. Now, the neurotransmitter, essentially released from the presynaptic membrane, is going to be absorbed by the dendrites. So there's going to be a chemical message that's going to go into the dendrites here that's going to start this whole process off. That's going to be transformed over time into what's called an electrochemical wave. So it's partially electric difference and it's partially chemical in nature. It gets passed down the neuron and ends with more neurotransmitter being released by the synaptic knobs of the receiving neuron. So this is going to happen very, very fast from one neuron to the next until a message is complete or shut down. To start up this idea, let's recall that all living cells maintain a difference in concentration of ions across their membranes. The overall charge of the interior is going to be negative in relation to the outside. Why is that? Well, it's because of, if you remember from the past, the sodium-potassium pump. Alright, so how does this work? Well, if you recall, the sodium-potassium pump pumped out 3 sodium ions for every 2 potassium ions that it pumped in. So what that means is that coming into this cell at any given time, we're going to have 2 K pluses, and coming out of the cell are 3 NAs. Alright, so we have a 3:2 ratio of NAs to K. Well, let's just arbitrarily throw some numbers out there. Imagine that 210 NAs were being pumped out. How many Ks would then be pumped in? Well, if we go by, if we take 210 and we break it into 3 parts, that would be 70 per part. So if 2 parts are coming in, that would be 140 potassium ions coming in. So there's a 210 to 140 ratio. Well, what's the difference there? Well, if you just subtract the 2, it would be about 70. So what that means is that the outside is 70 more positive. There's 70 more positive charges on the outside than the inside. So when we take a look at the charge of a cell, we compare the inside to the outside, and the inside tends to be negative 65 to 70 millivolts in comparison to the outside. So this middle of the cell gets a big negative sign, the outside of the cell gets a big positive sign. That's what we call our membrane potential. And we call it a potential because it's a type of stored energy. And as you know from middle school forward, potential energy is stored energy. And in this case here, all that abundance of positive charge on the outside is like water behind a dam. And if we're able to let it through, we can then power something else with those charges falling inward into the cell. Alright, so the magnitude of a potential difference between 2 sides of a polarized membrane is measured in volts, or in our case here, millivolts. What happens is there's going to be an electrode placed on the inside and an electrode placed on the outside, and then you measure the difference in voltage across the two. So a polarized membrane, what we're referring to here, is the fact that it has a positive and a negative side. And again, we said negative was in the inside, positive was on the outside. That information is going to help us understand the action potential. Okay, let's start taking a look at the action potential. So first thing, let's recognize that there's an ionic imbalance, and that is going to maintain your RMP, your resting membrane potential. Remember, I said that we place one electrode on the inside, one electrode on the outside, and we measure the difference. And from right here, you can see there's a ton more positive charges on the outside than there are on the inside. And what that's going to lead us to is a negative 70 millivolt difference across the membrane, measured by a voltmeter. We need to mention what a local potential is. I know there's a lot here, so we'll take it slow. Local potentials are slight shifts from the resting membrane potential in a specific region of the plasma membrane. Alright, so let's imagine that we have one nerve cell here, and my artistic ability is absolutely terrible. But we'll say this is one nerve cell, and it's sending a message from the left to the right. Now, our second nerve cell here, we said that there's dendrites all over the surface. And it's also pointing to the right as well, and we'll make its synaptic knob down there. So what's going to happen is that when the stimulus is released from the first cell here on the left, it's going to usually be by way of what's called a neurotransmitter, which are just little tiny chemical substances that are given off by the cell on the left and picked up by this cell on the right. And what it's going to do by landing on these dendrites is it's going to very briefly change the ionic imbalance that we see in this nerve cell. So this happens when a stimulus, both chemical or sensory, opens stimulus-gated Na channels. Now, a few slides ago, we saw the surface of a receiving dendrite. And what it looked like, and it was a real blown-up version of it, was something like this. There were little tiny proteins all over the surface, and these proteins caught chemical stimuli from the synaptic knob of the cell that is essentially allowing this chemical substance to leave it and go into the synaptic cleft. Now, in an excitatory stimulus, Na ions rush into the intracellular fluid through the channels. Those are these things right here, okay? So these respond, and when they respond to a chemical stimulus, they're going to pop open, and they're what's called sodium gates. And these sodium gates, they're going to open up, and they're going to allow all that sodium outside the cell to come rushing in to the cytoplasm of the receiving cell of that message. Now, if the interior of a cell is negative, what's going to happen is as those positive cells rush in through the open gates, they come in, what we see is that the inside won't longer be so negative as it was before, because you have positive sodium ions flowing into that. So if I have a whole bunch of pluses that are going into this cell here, that's going to bring that membrane potential down from such a huge difference. When that happens, it's called depolarizing a membrane. When something's really polar, it means that there's a positive charge and a negative charge and they're really far apart. But if you depolarize that difference, you're going to make it less so. So in this second cell, there's no longer going to be a negative 70. By having positive come in, it's going to go down negative 68, negative 66, negative 64. Depending on how strong of a message that is, will determine how low this may go. So maybe, it's a very weak message, and maybe it brings it down to about negative 62 millivolts. That's what we refer to as a local potential. It's a very small change in polarity due to the receiving of a message from a cell that's trying to propagate a signal. It's going to release its neurotransmitter. Let's say you're the neuron that's going to catch that particular chemical. You catch it, your sodium gates fling open, and sodium rushes in to your cell. Your cell is going to be depolarized. It's going to make your intracellular fluid, your cytoplasm, more positive than negative 70. So if you think about what's more positive than negative 70, well, negative 68 is, negative 66 is, negative 64. Those are all more positive because they're headed towards zero. Let's take a look at that scenario in a much better picture than I can draw. So here we have our nerve cell, and here we have those sodium gates we were talking about. Currently, we have one electrode on the outside, we have one electrode on the inside, and they're measuring out at resting membrane potential of negative 70 millivolts. Now, let's say that they receive a message from another cell. What's going to happen when they receive that message is these two sodium gates, and I'll put Na on them, they are going to open. And they're going to open in response to a stimulus, probably coming from the last neuron in the chain. And they're going to open up. And what we're going to see is we're going to see sodium ions rushing in. And what they do is they depolarize the membrane difference, in this case to negative 65. And if that stimulus is continued or it's very strong, these sodium gates will continue to stay open. And you'll have more and more sodium rush into this nerve cell. And over time, that depolarization is not just going to stay right there. It's going to start spreading. It's going to spread along the interior of the membrane. You're going to have some of it spreading to the right. You're going to have some of it spreading to the left. And as those sodium ions spread to the right and the left, what we're going to see more of those sodium gates popping open over here and over here. So now from our initial spot, we're down to negative 60 millivolts. And as that message goes to the right and continues popping open more sodium gates, this one's at negative 65. The message hasn't yet reached this one, which is still at negative 70, but it's next. So the stronger messages, or the more repeated a message is in a particular area, will pop open more sodium gates, allowing that sodium to rush in. There's also something called an inhibitory stimulus, which is going to open only the potassium gates. That's going to allow just potassium ions to flow out. That's going to make your membrane potential more negative than it already is. So in the case of a depolarization, that was excitatory, and it depolarized the membrane like you see here. So this made it go more towards zero. But in a hyperpolarization event, what we see is that only the K gates open, which allow for it to go the other way. So instead of going towards zero, maybe we're at negative 80, maybe we're at negative 90, which is essentially going to make it hard for a nerve cell to fire, which we're going to get into in just a minute. So a hyperpolarization takes it even further from zero. It makes it more negative than it already is. And what we're going to see is a lot of messages inside of a brain are going to have both inhibitory nature and sometimes excitatory nature at the same time. And sometimes the coordinated effect will produce the response in a nerve cell or a nerve pathway that we're focusing in on. Here's an example of a hyperpolarization event. Let's say the cell has some kind of receptor on its surface. I'll just whiten in a receptor right here, and we'll call this a receptor. It's terrible art, but we'll deal with it. And we'll say that some kind of neurotransmitter nestles itself into these particular protein channel membranes. But it has no effect on anything else. In fact, your sodium channels stay closed. You can see them right here. They're shut. They're not going to let any sodium from the outside in. But what we do see is because this neurotransmitter nestled into this membrane, this plasma membrane protein, it allowed for K gates to open. So there's sodium gates and there's potassium gates. And when K gates open, what's going to happen, because the interior of a cell is full of potassium ions, they're going to start rushing out. Well, isn't this area out here, isn't the concentration of sodium ions very high? Absolutely. So we're sending out more positive charges with the already positive exterior, which is going to make the difference between outside and inside even more pronounced. Instead of negative 70 millivolts, now we're talking negative 80, negative 90. It pushes it further and further away from stimulating this nerve cell if you have a hyperpolarization event. Okay, there's my arrows. A little late on the arrows, but hopefully you get the gist of that. All right, so the nerve impulse, we also call it the action potential, is an electrical fluctuation that travels along the surface of a neuron's plasma membrane. And it's usually the dendritic portion. Occasionally there will be some synapses on the cell body or soma as well. So let's take it one piece at a time. We say that this dendrite right here is sitting at resting membrane potential, which is negative 70 millivolts. And what we're waiting for now is possible stimuli from another nerve cell to bring our resting potential to a different spot. Let's see if that happens. Okay. So in step 1, an adequate stimulus triggers stimulus-gated channels to open. That's what we were talking about with those sodium channels. So they open, and that's going to allow sodium to start rushing in to this dendrite of a receiving neuron. That's going to produce what we call local depolarization. So if we're sitting at negative 70 down here and we allow sodium to rush into the negative interior of the cell, we're going to see that resting potential start to go up. No longer are we at negative 70. Maybe now we're at negative 60. But if we have a large enough stimulus, what's going to happen is we are going to exceed a certain point, what's called a threshold potential, which happens at negative 59 millivolts. When that happens, it's going to allow all of the neighboring sodium gates, the sodium channels, to pop open at the same time. If more sodium gates pop open, you're going to have more sodium rushing in, and that's going to further depolarize the membrane. So we're going to see it hit that negative 59 mark, and then it's going to take off. It's going to skyrocket because all the channels around it, all the membrane proteins, are all going to pop open at the same time, or at least close to the same time. So an excess of positive charges now rushing into the cell, depolarizing that cell. Voltage-gated sodium channels will stay open for about 1 millisecond until the cell interior reaches plus 40 millivolts. So what we were talking about last time, as so many sodium channels opened up, we shot all the way up past 0 to plus 40. Now at plus 40, what we're going to see is that our potassium channels are going to be triggered to open up. Because if you think about it, we've just made a hugely positive interior of the cell. Those positive charges are going to repel against each other. They're going to chemically pop open your potassium channels, and now potassium is going to rush out of this cell. And when potassium rushes out of the cell, what we're going to see is a loss of this positive exterior. We're going to reduce some of the repulsion that's going on here, the repelling nature of these positive charges, and that's going to send that change, that membrane potential, back down towards 0. And eventually it will reach 0, and it's even going to go further than that. So it's going to begin to move back towards resting membrane potential, towards negative 70 millivolts, where it normally is when K channels open. So the K rushes out all of a sudden. And remember, this spreads all over the neuron. When you have that happen, we call that the repolarization of the membrane, because negative 70 is a polarized membrane. To repolarize it means to put it back to negative 70. There is a brief moment of hyperpolarization. Because so much K rushes out, you're actually going to overshoot a little bit. And what will happen is that will get restored back to negative 70 by your sodium-potassium pumps. So if I continue this line here, it may actually dip even lower down here before it gets back in the right line. So if this is negative 70 millivolts here, maybe it hits negative 90, maybe it hits negative 85. But your sodium-potassium pump, with its 3 NAs out for its 2 NAs in, will eventually restore it, and it will go back to the same level again. Alright, so let's take it through. I like this diagram quite a bit. Let's go through it step by step. Alright, so first we're sitting at negative 70 millivolts. At this point here, we have the sodium-potassium system working. The pumps are going, 3 out for every 2 in, and suddenly we get a stimulus. That stimulus is going to pop open sodium gates. We'll say it's a strong stimulus. So all around that particular area on the neuron, the sodium gates everywhere are flinging open, and sodium is rushing into the cell. That depolarizes the cell membrane of the neuron. When it reaches a peak up here, what we see happen are the sodium gates slam shut. The interior of the cell is now, in this case here, it'd be positive 25 millivolts, but it can go all the way up to 40. That triggers K gates to open because there's so much positive charge in the interior of the cell. As the K gates open, K rushes out of all parts of that local area, and that's going to rapidly bring down the depolarized into a repolarized area, which means that we're going to get, again, a negative interior to the cell. It's going to even overshoot by a little bit and be hyperpolarized for a few milliseconds before it bounces back, because at this point your sodium-potassium pump, which will continue to work, will bring it back to normal. And at normal place, we'd have just the sodium-potassium pump working and continuing on at -70. After the action potential occurs, there's what's called a refractory period. This is a brief time where a local area of a neuron's membrane resists restimulation and will not respond to a stimulus no matter how strong. So this is going to be kind of a, once you send an action potential through a particular area of a neuron, it cannot be restimulated no matter what. We also can say that these messages that are moving very, very quickly down the dendrites, through the cell body's membrane, and then even onto the axon, they are going to be one-way transmission. And it happens very fast, and it can go even further if we have what's called saltatory conduction. Saltatory conduction is when we have that nerve impulse go down the axon here, but it doesn't need to stimulate every single sodium-potassium gate along the way, because this thing is currently wrapped in that insulating myelin. And we only have exposures right here in the nodes of Ranvier. So the actual message can jump from node to node to node much quicker than if it's just a bare naked axon without any myelin sheathing. How much faster again? 10 times faster. So it's going to run down this axon at an extremely fast rate, only opening the gates at the nodes where they're exposed. Here is what it looks like on the interior of that action potential that's wrapped in myelin, and the fact that you have your sodium and your potassium gates that are popping open in these two pictures here. Notice the sodium runs down the axon here to pop open a few more gates here, and you'll also see that the K, those gates will open in response to it, and it's going to travel this way. So it's going to be a wave of depolarization to best characterize the action potential. It's going to move in all directions, and specifically when we're talking about dendrites in the cell body. But then when it gets to the axon, it's more or less a one-way once that nerve signal has been kind of given the green light to go by the axon hillock. This is a picture of that refractory period we were talking about. Just a quick mention. This absolute area here, while the sodium gates are springing open, this is a point in which you cannot re-stimulate to re-pop open the sodium gates. There's a relative period, and this is when the K plus gates are popping open. It's going back to normal, the resting membrane potential. If you had an extremely strong stimulus, you can get this to once again pop open, but it takes a very strong stimulus to do so. And then once it's back to RMP, there's no problem in popping open the sodium gate again the second time. There's some great animations of nerve impulses. I tend to show my classes a ton of McGraw-Hill visualizations. I think they're great. So if you type in McGraw-Hill into your favorite search engine, and then the nerve impulse, or in this case here, voltage-gated channels in the action potential, you get a much greater look at the process that I can draw. I have terrible art skills again, so these animations are made by professionals. They are absolutely wonderful. I also like a really nice job done by someone at Harvard. I use this to show my classes as well. I'd pause the video, jot down this address there, pop it in, and follow along with the animation. It has you doing some stuff, and it's a bit longer, so please give it a try. You'll learn a ton by doing it. Okay, so the meeting place of two neurons is then going to be considered a synapse. There's two types. There's an electrical synapse, and that is where we have gap junctions allowing ions to flow through holes from one cell to another. So it simply is going to continue along postsynaptic membrane. The chemical synapse, however, is where we saw those neurotransmitters being released into the synaptic cleft, possibly inducing an action potential. Does it always happen? No, but that is the place where it happens. It's called a chemical synapse. Here is a picture of an electrical synapse. You could expect this in a place like the heart, where ions are going to just easily flow from one cell to the next through what we learned a long time ago called the gap junction. There is not a whole lot of room between the two, which allows these gap junction, these protein membranes, to allow flow. And if you have your whole heart being connected by ions, then the whole thing can beat at once without, your left side of your heart beating at its own time and a certain group of cells in another place beating on their own. They're all connected. So the heart is going to work as what we call a syncytium, a collective unit that carries a common function. Again, another picture here of electrical versus chemical. Here we have those gap junctions we were talking about where we have complete flow through of ions from one cell to another. But here we have a synaptic cleft where we have neurotransmitter that's being released as a number of vesicles being caught by the receiving neurons membrane on their protein receptors, and this may open up sodium channels. It might be inhibitory and open up potassium channels. There's a lot of potential for what neurotransmitter and what membrane protein can do, depending on the chemical message they receive. The anatomy of a chemical synapse, we've kind of gone over this terminology before, but let's just review a little bit. At the end of an axon, you're going to find a knob or knobs that are referred to as synaptic knobs, and we also call them axon terminus as well. And notice that they're filled up in this case with synaptic vesicles. So these are those transmitters, neurotransmitters that were produced. They came down a cytoskeletal highway, and now they're sitting in the end in the synaptic knob. And what's going to happen is they're waiting for a signal to release into the synaptic cleft, and then these neurotransmitter receptors are geared to catch that neurotransmitter. The synaptic cleft, again, is the space in between two neurons, and it's just an open area of extracellular fluid. Now, action potentials, they cannot cross the synaptic cleft in a chemical synapse. So it's going to continue its impulse by releasing neurotransmitters at the presynaptic membrane. These, we said, can be excitatory or they can be inhibitory. When an action potential reaches a synaptic knob, it's going to open calcium gates. So the electric potential difference that eventually makes its way down the axon is going to pop open calcium gates on the synaptic knob, on this axon terminus, and that's going to be the signal for these vesicles then to dump their contents into the synapse. So they're then going to be in the synapse area, synaptic cleft, and synaptic cleft, and they're going to then diffuse out towards receptors that are geared to catch them. So again, we have receptors on this postsynaptic neuron surface here. They're going to catch those vesicles and do something with them. Okay, so let's get into some more terminology here. We have the opening of ion channels that produces what's called the postsynaptic potential. We said it can be excitatory or inhibitory. So that's where these two acronyms come from. We have an excitatory postsynaptic potential, and we have an inhibitory postsynaptic potential. The EPSP, the excitatory one, occurs from the Na sodium gates opening. Okay, so that is that typical route where Na, the sodium channels, pop open first. Now, the IPSP, the inhibitory postsynaptic potential, these are going to occur because typically the K gates will only open, or even chlorine gates that we can find on the outside of neurons. When they pop open, again, we're going to lose that negative, we're going to allow chlorine from the outside to rush in, making the interior more negative. So again, in both cases, you'll see hyperpolarization occur. So here is an EPSP typical when you get a nervous signal. If it is excitatory, it's going to pop open sodium gates, which we can see there. If it's an IPSP, it's going to open either the potassium gate or chlorine gate, further polarizing the membrane into a hyperpolarization event. If it hits that negative 59, that magical number, you're going to have a full-blown action potential. Again, we'll get down, we'll go up. K gates will open up here. Na gates open here, and it will repolarize due to the sodium-potassium pump there. All right, couple other things that we can mention here during a synaptic event is that often we'll have what's called reuptake, and reuptake can allow released neurotransmitters to be cleared out of the synaptic cleft, and it can be taken up by various enzymes. It can be diffused and picked up by nearby glial cells. Like if there's a ton of neurotransmitter here and it's diffusing out, you might overwhelm these cells here on the postsynaptic membrane, and instead it keeps diffusing out and there will be a glial cell to take up all the extra. Or possibly you may have some reuptake occur where the same membrane that released it again absorbs it back in again. There are some psychoactive drugs that work to keep certain neurotransmitters in the synaptic cleft longer, and those are typically called reuptake inhibitors. Okay, so one of the ways that we can describe the scenario around multiple nerve cells releasing their message onto a receiving cell is through summation, which is also known as integration. Because one nerve impulse from one cell is not going to be enough to generate an action potential. Thousands to hundreds of thousands relaying their message is really going to cause that action potential to continue to propagate down a chain of neurons that have a function. So there's two types of summation that are out there. One is called spatial, and that's when you have multiple presynaptic membranes or synaptic knobs around a receiving cell at different places. So if my head is a receiving cell and my fingers are synaptic knobs and I have tons of them around my head, well, they're all giving the same message, and my head is then catching those messages and it's going to add together, it's going to summate, and if it's strong enough, it's going to pass it down the axon of my cell. Okay? There's also temporal summation. Well, actually, before we get to temporal, let me just mention again the axon hillock, and that was the portion of the neuron that was just between the cell body and the axon itself. So this region where it tapers down here, this, if the message is strong enough and they all summate in this area right here, that will allow that action potential, that nerve impulse, to continue, zip down the axon, and then release neurotransmitter when it reaches to the end. There's also a type of summation called temporal summation, and that's when you have not hundreds and thousands of connections onto the receiving cell, but much less. But those neurons keep firing over and over and over. So it's think of the part of the word tempo. The tempo is very high. It continues rapid fire succession to blast that receiving cell with messages, and that can eventually over time lead to an action potential being propagated. So here is an example of spatial summation. We have in this case one stimulus over in this part of the cell, one stimulus here, maybe we got another one here, another one here. Remember, it's going to take hundreds to thousands of these working on a nerve cell to eventually produce a high enough change in the polarization to hit that threshold potential of negative 59 millivolts collectively at the hillock to continue that message that way. So stimulus 1, you can see here, is it's pretty minor. Stimulus 2 is pretty minor as well, but the two of them together, and we'll say they represent a couple hundred neurons apiece, eventually are strong enough to hit that negative 59 and send this into full action potential, which allows that message to continue. Temporal selection then, let's say again we have a scenario where in this case this particular neuron here keeps firing over and over and over again. So if that happens, we eventually will summate all those messages here into an action potential. Their collective summation will allow it to fire, and our message will propagate down that axon. Now, sometimes we get in kind of a war between excitatory messages and inhibitory messages. So in this case here, if each neuron represents hundreds of neurons, well then we could say, okay, how many greens do we have? We got 1, 2, 3, 4, 5. How many inhibitories do we have? We got 1, 2, 3. So in this one here, because there's a lot more excitatory than there are inhibitory, maybe their collective energy, their collective messaging of this receiving cell is going to be more than what these red inhibitory ones can add to the mix, and because of that it hits its negative 59 and that action potential will propagate down the axon. As far as synaptic animations, again, I really respect McGraw-Hill's animations they put out there that are done by professionals. There's one, if you Google chemical synapse in McGraw-Hill, you'll get this one. It's great. Action potential propagation unmyelinated axon. Try this one as well. And here's another website that I really like. Pause the video, jot it down, give it a try, and it's a real nice one for synaptic transmission. So, a lot of information there. I hope that I helped you at least gain a picture of what's going on inside of all of these firing neurons that we have inside of our brains. It's complicated. It takes a lot of attention to detail to really get it. But as long as you do what you're supposed to do, listen to your lecture, back it up with textbook readings, do personal outlines, try to get these things straight as to what's moving and shaking inside of these nerve cells, and then hopefully you'll get a clearer picture of how cell communication and nervous system truly happens. So I hope this helped you understand a little bit better. I'll catch you next time. Bye.